with a nice American accent. Yeah, welcome to you all to this IBH seminar, another in the series, which has been really good. So, and this one, I'll welcome the three people who are going to speak. It's a triple act this time. The title they have is Barley, Running the Disease Gauntlet and Chemical Attack. How can we use barley genetics to help control disease? And for this, we've got, say, three speakers. Neil Havis, first of all. Neil's been probably known to many of us because he's been around quite a few years now. 30, I think 30 or so years, and a good 20 or so of those years working on perhaps what he's best known for, which is ramularia work. And very early on, ramularia and SRUC's work in this area became really well known in really establishing the epidemiology. And that's been of benefit to many, many people. Neil also has been heavily involved in the research work over the years, working on more sustainable crop protection programs. And he's also well known for all the work he does on AHDB projects, which are reported in various workshops and things of great practical value to many people. So he's also been involved in lots of linked projects, so well practiced in many practical applied projects as well as some of the more fundamental ones as well. The second speaker will be Francois Dussar, who he is a lecturer at SRUC, the same group, the Carbon Crop and Soil Group, which is now called IC. He's worked on plant pathogen interactions, and his PhD, which he finished in 2014, was on secondary metabolites in ramularia. And I know him from a project we were both involved in there a couple of years ago. The third speaker is a PhD student in her final year now, Laura Rurig, if I pronounced it somewhat near right. And she's working on control of ramularia leaf spot by plant genetics, a project that involves a real mix of bioinformatics, controlled environment, and field evaluation. So quite a wide range of things, and that involves Hutton, SWRI, and Edinburgh University, as well as SRUC. So let's get going with hand it over to Neil to share his screen and start this off. And please, as and when you think of questions, do put them in the chat and we'll come to them at the end. So over to you, Neil.
Okay, thank you, Adrian, for the introduction. Good afternoon, everybody. It's nice of Adrian to say it's 30 years. I think he's putting a few years on me, but never mind. I'll let him off with that. So I hope this works. And he gave me a build up and saying I've done lots of work on ramularia. And of course, I'll not be talking much about ramularia. I'm going to leave that to my colleagues to talk about that. I don't know, can you see that screen or not? Okay, that's great.
We can see that.
Okay, excellent. Good stuff. Okay, so hopefully, right.
It seems Neil has disconnected.
Yeah. I think he, not only his PowerPoint disappeared, but he disappeared.
Yes.
Okay. Can I suggest then that we, would it be okay if Francois, if you?
Yeah, sure.
Follow, yeah, probably it.
Yep. I'll share my screen. Hopefully the same thing will happen.
Well, hope not.
Okay. All right. Normally. Okay. Hopefully everybody can see that.
That's great. Thanks very much,
Perfect.
Francois.
Okay, so yes, good afternoon, everybody. And so I'm going to talk a bit about some of my work that's basically focusing on understanding how Ramularia produces a toxin that it supposedly uses during pathogenesis, although the actual mode of action or the role of the toxin currently is not fully known. And so without further ado, I'll start there. If you have any questions, feel free to ask them at any point. I'm happy to be interrupted if you have questions about that. So first of all, starting with what are rubulins, just if you don't know what they are, so they basically are secondary metabolites. In other words, they're toxins that are produced by Ramularia colosigni. Although they've been identified in Ramularia, but the first organism from which they were identified is actually Mycosphaera rubella, which is a pathogen on different plants, not at all on cereals actually. So quite interesting that this pathogen also produces rubulin. And these toxins, I call them rubulins because there are several derivatives, but mostly in plants you only end up with 2 derivatives, and particularly rubulin D. And these toxins, all of them, all the rubulins are photoactivated and they're non-host specific. So what that means is that if you were to take leaf, for instance, here you've got barley leaves that were cut and they were basically let to soak in a solution of rubulin. And just like the leaf would basically just absorb the rubulin. And if you put them in the dark, obviously, as you can see here, you don't get any symptoms. You do get a bit of coloration, but that's just because the toxin is actually bright red. And if you put it in the light, then you end up with some necrosis, as you can see here. So that's what light activated means. And also they're non-host specific, so they basically kill any type of cell they get in contact with. Sorry. And you've got the example here with tobacco leaves that have been infiltrated with the toxin, and you can see cell death going on in those leaves as well. And why do we focus about the rubulin is because we know that the disease symptoms has been associated with light levels. There's a few authors that have shown that, whereby if you have increased light level, you tend to have increased level of disease severity. So this about the link that we know between rubulin and disease level, but we don't really know exactly what the toxins do in terms of disease development. So my work focuses on understanding how are the toxins synthesized, and that way we're trying to understand basically the nature of the link between rubulin and Ramularia leaf spot. And I also have a part of work where I look at how the toxin actually works in the plant. So that's something I'm not going to talk about today. I'm just going to focus on the rubulin synthesis today. And some of you might be a bit familiar with some of this work that I've presented before as well. So before I started, the first thing I did was to look at the toxin and look at the chemical structure of that toxin. So you've got rubulin C and D here, so fairly big molecule. But if you look at it closely, you actually see that it has a dimeric structure, and it's actually a dimer of chrysophanol that you've got here. And if you're not convinced by looking at it, because, okay, you've got one subunit here, the rest is a bit more difficult, look at this molecule here, the original rubulins, which are known to be produced by Ramularia as well. They've been identified in Ramularia cultures, and we can safely assume that these are intermediates in the production of rubulin. And here you can really clearly see that dimeric structure with one subunit of chrysophanol here and another one there. That one's slightly modified, but only a few chemical modifications are needed to go from this to that. And this dimeric structure is something that's actually quite common, particularly in closely related organisms. And this has been shown in Cladophylvine fulvum, which is a pathogen of tomato. It's closely related to Ramularia, and it produces a toxin called Cladophylvine, which you've got here. And you can see here, it's a bit more obvious. Again, a dimeric structure, but this time it's a dimer of nataloimodin, which is closely related to chrysophanol. And the good thing with these is that both chrysophanol and nataloimodin they have common intermediate in their biosynthesis, and they all have this molecule here, which is called monodictyphenon, and that's produced by Aspergillus. So to identify basically the genes involved in the production of rubulin, I started with the assumption that the set of genes required to produce rubulin would require the production of chrysophanol, and therefore they would have some form of similarity with the set of genes needed for cladophylvine or monodictyphenon biosynthesis. So what I did is basically look in the genome of Ramularia for every single potential gene cluster that I found, so basically set of genes that are associated with secondary metabolism. I tried to identify some that showed similarity with known secondary metabolite biosynthetic cluster in other organisms. And there's three that sort of jumped at me very quickly. The first one is the top one you can see here. Basically, at the top, you always have the Ramularia cluster, and at the bottom, you've got the other species. So in this case, it's Phoma-beta. So it's the cluster responsible for the biosynthesis of beta-enon in Phoma. And Phoma-beta is a pathogen of beetroot, and beta-enon actually induces chlorosis in beetroot. And you can see those dotted lines actually are showing the similarity, the homology between genes in Ramularia and genes in Phoma. And you can see that the genome, at least in the genome of Ramularia, you have a full gene complement necessary for the production of beta-enon. There's a few other genes. We don't know if they're expressed. We don't know, there might be pseudogenes. We don't know what they do, but at least there is the enough genes or the full gene complement to produce beta-enon or some form of derivative of beta-enon. You've got the same thing for the bottom one here that I'm going to explain first, is the cluster responsible for chitoglobosine A production in penicillium, and that chitoglobosine is basically an antifungal agent. And again, if you look at this, you can see that the Ramularia has, again, all of the genes necessary for chitoglobosine production and an extra few other genes. Again, we don't know if they're expressed. We don't know if they are real genes or pseudogenes, but at least Ramularia seems to be well equipped to produce some form of relative of chitoglobosine. But really, the gene cluster that we'll focus on today is the one in the middle here. It's the one that exhibits similarity to the monodictyphenon cluster in Aspergillus. And you can see that there's, again, a lot of similarities and a lot of homology between genes in the Ramularia cluster and genes in the monodictyphenon cluster. There's a few genes that are missing, and I'm going to focus on the MDPL right there and MDPD right there. So these two genes really are crucial in the biosynthesis of monodictyphenon. The other two that are here, MDPI, MDPJ, these two genes aren't involved in monodictyphenon biosynthesis, but they're involved further downstream, so involved in something after monodictyphenon is synthesized. So these guys, not too worried that they're not present in the gene cluster, but MDPL and MDPD were somewhat of a problem that we didn't find them in this cluster. Nonetheless, we had already quite a good set of homologue and a good idea that this cluster might be involved in a production of at least of an anthraquinone toxin, something related to Rubling. So if you remember what I mentioned before, saying that Rubling and Cladophylvine, or at least chrysophanol and nataloimodine, were closely related, I started thinking, well, this cluster, it would be worth to look at that cluster and compare it to the cluster in Cladophylvine. And that's what I've done next. Is basically looking, so at the top here, you've got the cluster responsible for cladophylvine production in Cladosporium fulvum. And the first thing you can see is that this gene cluster actually is not just one cluster. You've got a main cluster here, and you've got two, what they call mini clusters. It's basically, in this case, single genes that are located elsewhere on the genome. And this might seem strange because the sort of accepted fact is that normally genes involved in secondary metabolism are clustered together. But actually, across a lot of different organisms, this sort of organization is not uncommon to have genes spread at different locations in the genome with a main cluster and several smaller clusters around that. And you can see that actually, when you look at the cladophylvine, every single gene in the cladophylvine biosynthetic cluster has a homologue in Ramularia, including this CLA-L, which basically is a homologue to MDPL that I mentioned earlier that wasn't present on the main cluster in Ramularia, but actually we found a homologue sitting outside of the cluster somewhere else. And the same goes for this MDPD that I mentioned before that's required for monodictyphenon production. This one also present somewhere else on the genome. So by doing this sort of extra analysis of looking at cladophylvine and looking more in detail as well outside of this cluster, looking more basically in the whole genome for potential homologues of monodictyphenon and cladophylvine biosynthetic genes, we ended up with a more complete idea of what the cluster might look like, with this cluster being organized around that main second metabolite cluster. and then having two small, again, mini clusters around it. And as I said, the idea that the co-location is important is something that used to be very important, very crucial for second metabolism, but actually nowadays the most important definition is not necessarily the co-location for genes, but the co-regulation of genes. So you need to have genes that are expressed at the same time. That's a sine qua non condition to be part of a gene cluster. So that was the logical next step, was to investigate these genes present on Ramularia and try to see if they were co-regulated or not. So I started looking at I basically used a study that was done back in 2016 when the genome of Ramularia was published. We had some RNA-seq data that were available, and it wasn't a very complete data set. It wasn't a very great RNA-seq data set because we only had 2 time points, but that was a good basis. And I started to compare every single gene present on this cluster to this one, this RCCG. The reason why I compared the expression of those genes to that one is because this is what we call the core gene. It's the very first gene involved in the production of the backbone, basically, of the toxin. So in the RNA-seq data, what I did is basically look at all these genes and compare whether or not they were expressed at the same time as RCCG. If they were expressed at the same time, I would consider them to be potentially co-regulated. If they were not expressed, I was basically discarding them from the cluster. And these are the genes that you can see that are basically painted in gray here. So these genes, based on the RNA-seq data, didn't appear to be co-regulated. All of the other ones potentially were. And I decided to then further investigate the other one by looking at their expression during disease development in plant. And you can see, if you look at the RCCG here, you see that it's highly expressed at 5 and 7 days post-inoculation, and then it quickly drops. And interestingly, all of the genes on the gene cluster exhibit somewhat of the same pattern of expression, with mostly the expression being there at 5 and 7 days and then dropping for all of these guys. So that's quite interesting in a way, confirming that these were all involved in that cluster, with the exception of RCCA, but that's a bit different because this is a transcription factor. So it might be the fact that its expression level is slightly different is not too surprising. I also investigated a gene that we knew wasn't co-regulated, just to confirm the validity of the RNA-seq data and of this approach. And you can see that indeed that gene exhibits a completely different expression profile, further confirming that all the genes identified in the RNA-seq data as non-co-regulated were indeed not part of that cluster. The interesting part with this expression analysis is that basically the 5 and 7 days post-inoculation is before we see symptoms appearing. So it seems that all of these genes are expressed before or at the early stage of symptom development. Okay. So by doing this, we ended up with the idea or a better idea of which genes really are involved in the biosynthesis of rubulin. And that allowed us to come up with a potential pathway, basically. And that's where my collaborator, Dorota Jakubczyk, comes into play. She's a biochemist. And based on the putative function of the genes, she basically was able to reconstruct the pathway leading to rubulin biosynthesis. And in this pathway, there's a few genes that really are key genes. The first one, obviously, is RCCG. As I said, it's the one responsible for the biosynthesis of the core structure, of the core of the backbone of the chemical. However, this one might not be really that key because it has been shown in other fungi that if, for whatever reason, that gene was to be basically knocked out or become non-functional, there are other polypeptide synthase in the genome of Ramularia that could potentially take over, and that has been observed in other fungi. So that could be a very interesting gene, maybe not necessarily the most interesting. Some of the really interesting ones are the RCCL. The reason why this is interesting is because of its nature. It's a Bayer-Villiger monoxygenase, and in the genome of Ramularia, there's only one of them. It's the only gene that does this. And if you look at other fungi, there's actually very few, a very limited number of Bayer-Villiger monoxygenases present in fungal genome. So that's quite interesting. And finally, the last one, the most interesting to me, is the RCCM. And the reason for that is because it's responsible for the dimerization step. And actually, that dimerization step in Cladophylvine it was shown that it really drastically increased the toxicity of cladophylvine. So if you were to take the monomer, is much less bioactive than the dimer. And we had the same idea, thinking maybe we should investigate that and compare the toxicity of the monomer compared to that of the dimer. So to do that, I've infiltrated leaves of Arabidopsis because the toxin is non-host specific. I basically infiltrated leaves of Arabidopsis with Rubling and with chrysophanol. And the picture basically speaks for itself. If you infiltrate chrysophanol at the same concentration, you absolutely get no symptoms at all, no cell death, very similar to what you would see in the control, which basically has no cell death. And even when you increase the concentration up to 1 millimolar, you still get a much lower cell death or a much smaller lesion there. And what's interesting to note as well is that the phenotype is very different between those two cases. So here you've got the typical dehydrated collapsed tissue, really typical of programmed cell death in Arabidopsis, whereas here you got a more water-soaked lesion. So that to me suggests that the mechanism also, the mode of action is also quite different between Rubling and chrysophanol. So as a conclusion, so at the end of that study, we basically had a very good idea of what the complete gene cluster for Rubling biosynthesis is, and it seems that it's spread across several loci in the genome, which again is not uncommon. We have shown as well that this dimerization step really seems to be crucial in increasing the toxicity of the molecule, but also on modifying its mode of action. And quick word just on the mode of action of the toxin itself. I've mentioned before that it looked like a programmed cell death in Arabidopsis, and actually some of the work I've done tends to really point towards a induction of cell death in plants through the induction of the host unfolded protein response. So basically you would then end up with an increase of unfolded protein, and that leads to cell death eventually. So that's basically, in a nutshell, my work. And again, there's still a lot of work that's needed to understand what the toxin really does in relation to disease development. But step by step, we're hopefully we'll get there. And that's the end of my presentation. If you have a question, I'm happy to take them. I'll stop sharing my screen. How do I do that?
Thanks, Francois. I think we'll go keep questions to the end. We'll go straight on to Neil, who is back, I see. Hopefully his PowerPoint will share straight away.
I hope
to you,
so.
Neil.
I must apologize for disappearing off just when I was asked to share my screen. Try again. Okay, I hope that's coming up. Can you see it now?
Yep, that's great. Thanks.
Okay, so what I'm going to talk about is running the disease gauntlet. So it's a bit more different kind of talk from Francois. If I can get onto this to change this. So I just wanted to make the point and talk about all the different diseases which attack barley. Okay, if you can move on next. Can you see the next pits? Has it moved on at all?
Not yet.
Not yet. Okay.
You might have to click on the. I had to click on the first slide, and then with the arrow it worked.
Okie doke. Ay, caramba. One of those days.
Neil,
Change
you
now?
said, oh,
Has it
So-
changed now?
all
Yes,
good.
Barley life cycle is there now.
Yeah. Okay. So that. Sorry about that. So what we're interested in and what we're trying to do, obviously, is get the barley crop through its life cycle. So we're talking about a plant, and we're talking about how we protect it at the seed stage, how we protect it as the seed starts to develop and germinate and form seedlings, and then through the development of canopy and the vegetative growth stage of the plant, and then of course we come to flowering. How do we protect it? What pathogens can possibly attack it at this point? And next, underneath that, of course, we've got the preservation of green leaf area. We know that barley is an example of a sink limited crop. So we need to protect it early on because that's when potential yield is set. So we need to try and keep disease out. But then, of course, we need to maintain that green leaf area to produce the photosynthate to fill the grains that you see in the nearly ripe crop on the bottom left of the slide. Okay, so that's what we're trying to do. And we're going to talk about some of the diseases which can possibly interfere with this whole process. So I thought I'd start with the barley seed, and I've titled this The Barley Seed the Enemy Within. And I'll give you some examples of a number of pathogens. And Francois he's probably touched on the Ramularia life cycle, but we'll talk about that. That's an example of a pathogen which is within the barley seed. So what we're trying to do is keep the disease out, and we're up against a number of pathogens which exhibit a number of strategies to try and infect the seed. Okay, so first thing you can see in the top left, this is some GFP imaging of Ramularia colosigni, and this was a PhD student which we had a few years back who did some work at Ramularia, and he actually, using this genetically transformed fungal cultures, he was able to look at the infection process and look to see where Ramularia gets in the seed. We knew from some DNA testing it was within the seed, but what Mathieu was able to show is it actually is under the alurone. It's under the seed coat, just outside the alurone layer. But if you can make out possibly in that image, there are some traces of that green fluorescent protein down into the further within the seed as well. And when we actually did some dissection, and other researchers did some dissection, they found it within the embryo section of the seed as well. And there's an image of Ramularia symptoms below, but I'll not talk too much about Ramularia because Laura's coming on next. To the right of that, we have the Eustulago, the smut species. And this is something which is intimately associated with the actual embryo of the seed. So it actually invades the grain sites during the flowering process. And what it does is then replace the grains with a whole mass of spores. These can break up, these can infect crops during harvest. If you don't have effective chemical control, and in some less developed countries, you get massive infections of grain, which is going to be used for seed the next year. And all you get is you don't get any grains, you just get these massive spores. Okay, so this is a fungus which is deep within the seed. It interferes with the final crop in terms of the grain. It also interferes with the plant physiology. So these plants tend to grow taller and mature faster. So you can actually see them, they stand out from a barley crop. Okay, something which is not actually within the seed, but would be sown with the seed. In the top left, this is Claviceps Purpurea. The common name is ergot in grain. This is another pathogen which infects the barley ear during flowering. What happens is spores spread up from some of these structures, overwintering structures. They infect the barley ear at flowering. They actually infect the ovary and produce something called honeydew, which is the sugary substance which actually attracts insects, and the insects will help move the pathogen from ear to ear. So that's a very clever fungus in terms of its how it moves itself around and propagates itself. Bottom right, Paranormal Triticis is something called leaf stripe. This is something, again, we've relied on chemical control. This is a pathogen which colonizes the barley seed coat, but it can build up from generation to generation. And if you don't control it within a few generations, you can have complete crop collapse and complete crop failure. So this is, again, something which can build up in the crop. These are two examples of major pathogens of barley, which can be found within the seed, but also in the trash. So things like crop debris and volunteer plants. The top left is Rincusporium commune. This would be the major pathogen we face in Scotland. It's the big yield robber. It infects early on. It causes significant yield loss. You can split pathogens into different groups depending on how their life cycle and the mode of action, how they live themselves. So we split them into necrotrophs, those which rely on dead tissue or feed on dead tissue, biotrophs, those which feed on a living plant, and hemibiotrophs, those which can exhibit both lifestyles. Rincusporium communis is a good example of a necrotroph, so you can see it's killing the cells around it, it's forming these lesions, it's killing the green leaf area. That obviously is going to have an effect on yield. There have been yield losses of up to 40% reported to Rincusporium. Okay, and the bottom right is Paranormal Triticis. This is net blotch. This would be a major pathogen in possibly some of the warmer countries. Anybody who's been around barley crops in Europe will have seen lots and lots of net blotch. And it is a major problem because, as you can see, it's also taking out the green leaf area that the plant needs to go through its life cycle fully. Again, this is an example of a seed-borne pathogen. It's also spread in trash. Most pathologists think that the trash-borne infection is more important than seed. This is a photograph taken from a spring barley crop in 2021 in Lanark. It's one particular variety, and I went around a number of trial sites last year, and it's the same variety. So the same seed stock gives you the same symptoms irrespective of the site. So it's quite important that we try and keep seed clean as possible. Okay, so there are a number of specialist barley pathogens. We've seen a few of them there. There are also a number of generalist pathogens which attack not only barley but other grass species and wheat. So on the left, you've got Septorium nodorum. This is a pathogen most commonly associated with wheat. This is a photograph from a bog hole trial site in 2001. And you can see the pathogen is taking out the green leaf area. It's affecting the crop growth. So Septoria is known to do that. It has a fairly warm optimal temperature of above 20 degrees and high relative humidity, and it can have a significant effect on yield. Paranormal Triticis repentis is again more commonly associated with wheat crops, but we are seeing this building up in barley crops. It colonizes the barley seed coat, and there are concerns about this building up and becoming more of an issue with barley growers as we go through time, because that photograph that's taken there is from a fully fungicide treated plot which controlled every other disease, and as you can see there, it didn't control the Paranormal Triticis repentis. So that's a minor pathogen at the moment, but one it's worth keeping an eye on. The bottom right is Cladophylvine graminarium. It's a seed and trash-borne pathogen. It also infects wheat crops, so it naturally infects the barley plant through any damaged roots. It moves up the stem. It actually blocks the vessels at the base of the leaf. So that distinctive yellow stripe you see up the leaf is caused by a blockage in the vessels within the plant. And actually it is an example of a true wilt. So those vessels are blocked and that leaf is wilting further along the leaf. So one of the major issues which affects barley crops as well, it's the enemy within, is Fusarium foot rot. And the Fusarium complex is a combination of Fusarium species and also Microdochium nivale. What you see at the base of the stem is brown lesions. So the pathogen colonizes the surface of seed, which is generally in Scotland, we always recommend seed is testing, so we know what kind of levels of infection we have. There are effective seed treatments to control it. If you don't control it, you can see the picture above Fusarium foot rot. That's an example of a Microdochium infection. In a cool, wet soil, it can attack the developing plants and it can cause plant death. Okay, so we're trying to avoid that. So this is a major issue. On the right-hand side, you can see some pictures of barley ears, and we'll have more about that later. But what happens is the fungus is spread up on the canopy by heavy rainfall events. The crop's particularly susceptible during flowering. You get conidia moving up and it can cause infections of the ear. And there are potential implications of that, and I'll come on to that in future slides. But this is an example of a pathogen which is not just present in the seed and trash, but it's also present in the soil. So here again, we're planting our plants into a potentially hostile environment. Eye spot is another example of a pathogen which is in the soil. It builds up in trash. It attacks a number of cereal crops. So even if you're trying to control it with things like rotation, it can survive in the soil for up to 3 years. So you'd have to seriously consider how long a rotation and how long a break from cereals you're going to have. And what you can just about make out in the top left, that picture, these are eye spot lesions at the base of the barley stem. And what they do is they weaken the stem. And obviously what we're trying to do is get a good crop, a healthy crop, one which is able to stand until it's ready for harvest. And if you can see in the picture of the bottom right, that's a picture of a crop which has gone down. So it's gone down under the environmental conditions like rain and wind, and it's called lodging, okay? And if we don't control the eye spot pathogen, you get increased lodging. So all the time, effort, and expense of producing a barley crop has been a complete waste of time because it's not harvestable, okay? Two other major pathogens which can severely affect barley production. Top left, this is a pathogen called Tacol. It's Guanamyces Graminus. It can go for barley. It's not as big a problem in barley as it is in other crops, but it produces a rat's tail. It can build up in the soil. Again, you need to either have a break from rotation, or sometimes you have to bite the bullet and continue to grow a susceptible crop such as wheat for a number of seasons to allow antagonists to build up in the soil and help control the Tacol. But to the right hand side, you've got another eye spot pathogen. This is sharp eye spot. What this does is actually attack, again, the stem base. So again, you've got weakened stems that can make the crop more susceptible to lodging. With the Tacol, if you get Tacol infection, you can see the roots are stunted. So there's limited transport of water and nutrients. And what you'll see in the crop are white heads. They don't look normal. And that obviously constitutes a significant loss in terms of production. So we've planted our poor barley plants into the soil. They've had seed-borne pathogens. They've been attacked from the trash. They've been attacked from the soil. And then, of course, you come down to the foliar pathogens. Blue Graminus, or powdery mildew, you can see the top right, that would have been the major barley pathogen 35, 40 years ago. It was the one which many spring barley varieties were susceptible to. But with advances in genetics and the introduction of MLO-based resistance for powdery mildew, we've almost eliminated that from being a problem for barley growers. In fact, the HDB current recommended list, there are no spring barley varieties which don't have MLO resistance. So that's one which we have used or practically got under control with breeding. It has an optimum of 15 degrees centigrade and it likes high relative humidity. It's still an issue in winter barley crops. Below that, you've got Petunia hordae, or brown rust. This is something that overwinters on volunteers. Spores are produced. And again, this is one which is considered to be an indicator of climate change. It likes warmer summers and high humidity. It has a higher optimal temperature, 15 to 25 degrees centigrade. So this concern is that Petunia hordae and brown rust is something we're going to see more epidemics of on an annual basis. To the left of that is a rather spectacular picture of Petunia striformis, FSP hordae. This is yellow rust on barley. So in general, it's only usually an issue if you have susceptible varieties, if you have a cool but mild winter, which allows the pathogen to overwinter. It's always considered far more of a problem in wheat than it is in barley, but that's a 6-row barley crop at our bog hole site last year in 2021, and you can see the leaf surface is completely covered with yellow pustules. Okay, so these three are examples of biotrophs. These are examples of pathogens which want to keep the barley host alive for as long as possible. They will even interfere with the physiological process of the plant to keep the plant photosynthesizing close to the infection sites. So more of a challenge for the barley plant to fight them off, but again, a different strategy to some of the other pathogens that we've seen. Back to the fusarium. So I mentioned the fusarium head blight, and its fusarium head blight is a complex of many different fusarium species and also the mycodochium. Why they're an issue? Well, many of the fusarium species are capable of producing something called mycotoxins, and these are harmful to mammals. They're harmful to humans as well. If they enter the human food chain, they can interfere with the processing of barley grains as well. It can cause issues in brewing and sometimes in distilling. And as a rule of thumb, you can almost differentiate between the fusarium species depending on the kind of symptoms they produce on the ear. So Calmorum and Graminarium would give a bleached ear. You could get spikelet bleachers, that's Mycodochium, generally Mycodochium species, or you can get Fusarium species which cause distinct blotches on the ear. And that could be things like Fusarium poe and Fusarium lanceti. So again, Fusarium is something with major concerns about. So there are limits and guidelines for the amount of mycotoxin that can be present in food substances going forward. But of course, if you're using barley for animal feed, those kind of things are not tested. So we'll have to be careful to try and keep them out of the production system. And last but not least, we've taken the barley through the whole growing process. You've protected it as much as you can, and then it's standing in the field waiting for harvest. And unfortunately, again, it's still susceptible. What you get is barley is under attack, and these are late season spoilage organisms, things like Alternaria and Cladophylvine, and they can even be attracted by the activity of insects. So things like aphid honeydew that can help spread these pathogens as well. Okay, so what they cause is discoloration. They can almost make grain unmarketable. So I hope you can see barley from its initial sowing right the way through runs a whole gauntlet of disease in terms of pathogens which can be within the host tissue, outside the host tissue, those which cause cell death, those which kill the cells, those which keep the plant alive. But the ultimate loss is production, because all of these pathogens will have an effect on the photosynthetic area and ultimately the yield of the crop. So it's important that we try and control them as best we can. Okay, and if I can get out that screen and stop sharing.
Okay, thanks very much, Neil.
If I can.
If you can
Okay, I'm out.
that's great. It's amazing anything actually survives with all that threatening it. Shall we move straight on to Laura, and then we'll come to questions immediately after that. So Laura. Yeah, that's brilliant. We can see it. But we can't hear you. I think you're muted, Laura.
Yeah.
Yeah, we can hear you now. Great.
Great. Oh my God. Okay, great. So yeah, I'm Laura. Thanks for still listening. My project, I'm in my final year, as Adrian just mentioned at the beginning, and my project is about trying to understand the genetic basis of Ramillaria disease resistance in barley. And now I'm having the same problem. Great. And Ramillaria, so Neil targeted already, that we have to protect barley from all sorts of diseases, and Ramillaria leaf spot is one of those annoying diseases barley gets attacked by. And currently, barley, or we can control Ramillaria leaf spot in barley mostly via fungicide, foliar fungicide applications. But the challenge with Ramillaria is that it has the ability to evolve fungicide resistance, especially in the single-site fungicides, and has done so already in the QI. And in Germany and the UK, there are also reports of fungicide resistance against SDHIs and DMIs. And further challenge in the control of Ramillaria came into place at the beginning of, or in the middle of the pandemic in 2020, when the most effective fungicide, the multi-site active chlorothalonil, was banned in Europe because of several reasons for the environment. And because Ramillaria leaf spot is a very late season pathogen, it's very difficult to predict it anyway to control it via fungicides. But to even increase the challenge for this disease, at the moment we don't know any varietal genetic resistance to Ramillaria leaf spot, and therefore my project comes into place, and the importance to investigate if there's a potential genetic control against Ramillaria leaf spot. And this is what I'm talking about today. So I'm basically trying to give you a brief overview about my project, what I've done so far, just very briefly, and yeah, and then feel free to ask any questions. So the first aim of my project was to see if there, or to characterize genetic regions against resistance to Ramillaria leaf spot. And I've done this by doing a genome-wide association study with the help of the James Hutton Institute. I used those findings to do validation experiments in the field in Scotland last year. And I'm planning to do controlled environmental experiments of those same varieties under controlled conditions after my second field season. And one of the reasons, or one of the problems with Ramillaria, is that we know from studies and from observations that the environment plays a crucial role, or one role, in the disease development, because Ramillaria has the ability to stay asymptomatic for quite long in the season. As I just mentioned, it's a late season pathogen. And something triggers the transition between this endophytic lifestyle to the necrotrophic development. And therefore, one of my second aims in my PhD is trying to understand this relationship better between Ramillaria, its environment, and the symptom development. And because senescence is one of the physiological responses to several stress factors, such as drought, or also developmental processes of the plant itself, such as seed development, we thought we want to see what kind of role senescence plays in this relationship. And therefore I've done an experiment where I'm trying to manipulate artificially the barley leaf senescence by 2 different phytohormones to then investigate how it impacts Ramillaria growth and symptom development. And these experiments actually led to a very interesting other experiment. So preliminary data suggested that by applying ethylene, which is actually meant to induce senescence artificially in barley, actually reduces symptom expression and Ramillaria growth in planter. So therefore, we thought it would be quite interesting to try to identify barley genes that are responsive to ethylene and to monitor the gene expression in those marker genes after ethylene treatment in different barley cultivars that also differ in the susceptibility to Ramillaria itself. First of all, I want to take you back to my first year of my PhD, when I've done, together with the James Hutton, the genome-wide association study. And for those who are not familiar with the smart word of GWAS, I try to explain this briefly to you. So basically, for a GWAS, you need a genotypic data set, which means we need a huge data set of single nucleotide polymorphisms, so-called SNPs, that have been published of a lot of barley varieties at the beginning of 2017. And what you also need is a phenotypic data set. And by phenotypic data set, in my case, I mean disease symptoms like Ramillaria leaf spot, as you can see them in the picture, of as many varieties that differ in their genes as possible. And I was lucky that I was provided by a data set from 2013. It was a massive project of huge collaborations where researchers have scored for Ramillaria leaf spot symptoms in over 260 spring barley varieties in Norway, in Germany, and in Scotland. And I was able to use the symptom data of this data set to see which of those varieties are matching the published 50K SNP data set. And in the end, I managed to get 239 matches, and I've carried out the so-called Gras analysis, which basically tries to link genotypic association variations to differences in the phenotype. So basically looking looking into if the genotype can explain the phenotypic variation. And what we then get if we run this analysis is a so-called Manhattan plot that you can see on the right-hand side. So on the x-axis, we can see the 7 barley chromosomes, and on the y-axis, it's basically the probability that this SNP is responsible for this association. And I've highlighted with a green arrow the SNP we found, or the quantitative trait loci we found. It's at the end of chromosome 4H, and it's an A and G polymorphism. So that was great. First year, first aim succeeded. It's not always like this in a PhD, I can tell you that. But what it provided me with is I had the QTL. I was able to use the data set then to look for the most susceptible and the most resistant spring barley varieties. And then, because obviously I can't test all 230 barley varieties again, it's humanly not possible, I think. I've decided to take the 10 most susceptible and the 10 most resistant spring barley varieties. But because I had the genetic region now, I was also able to identify candidate genes that are laying under this QTL locus. But yeah, as everything in research, we need to validate our findings. So what I've done last year is I've run 25 spring barley varieties in Germany and in Scotland. In Germany, unfortunately, we didn't get any symptom data. Ramillaria just didn't want to join the party there. But in Scotland, we managed to get nice disease symptoms. So I was scoring for Ramillaria post-flowering when the symptoms typically occur. And I've also monitored Ramillaria DNA levels in planter. And because we, as I mentioned before, we want to better understand the transition between asymptomatic growth and then the necrotrophic development, we decided to monitor DNA level at 2 asymptomatic growth stages and then at the scoring stage. And just to take you with me to the field, I've taken the most susceptible and the most resistant lines of taking pictures of. So Chieftain and Hydra were the ones with the highest disease symptoms, whereas RGT Planet, which is known from literature already that it's quite resistant to Ramillaria, and Aramir, it's from my GWAS analysis, which is quite resistant as well. And if I just briefly show you my results from my field trial. So what I've done, or what we've seen, or what we are seeing here, are the 14 different barley varieties, and 14 is because some of the varieties were not germinating. And in yellow, you can see the susceptible barley varieties that are carrying the G allele of the QTL. And in green, you can see the more resistant varieties that are carrying the A allele. And what was nice, or what is nice to see in this graph, is that actually those that were named susceptible varieties were again in Scotland quite susceptible, and those that are meant to be more resistant based on the QTL were again more resistant. And I know there's in the middle, there's a little bit of a mixed picture, but we know it's never like a fully resistance or a full susceptibility. Therefore, we are looking to find a quantitative trait loci. And on the right side, I've tested some barley varieties that are known from literature to just compare our conditions to their conditions, and varieties where we kind of know that they are more resistant or not, like Power and Bremer, for example. And it was quite nice to see that Power, which is quite resistant against Ramillaria, was again under the conditions quite resistant, and Bremer was more susceptible. And the same for RGT Planet. What was interesting is that I've chosen Scarlet, for example, from the German paper that was published last year by Hollander. It's a German lab. And in their field trials under irrigated condition, it was actually one of the most susceptible varieties, but under our Scottish conditions, it was a quite resistant variety, which is very interesting and highlights again the importance to investigate what relationship the environment has together with Ramillaria and barley. That brings me to my current and future work. So as I mentioned, I will take these findings from last year, but also from my season this year, to do controlled environment experiments. And I will have a second field season in Germany and in Scotland again. This year, I will have a different site in Germany provided by KWS again in the south of Germany, which is known to be more likely to get Rammelaria. And I will be again sampling at the asymptomatic growth stages to get a better picture, and will be scoring both in Scotland and in Germany myself. And I'm also planning to do some spot measurement of a subset of varieties to further test the link to senescence. And while we are at senescence, I just briefly, before I finish my talk, I wanted to show you my my latest data of my manipulating barley leaf senescence experiments. So as I mentioned before, I used ethylene, but I used an ethylene precursor, ACC it's called, to see how it impacts Rammelaria growth and disease symptom development. And what I've seen after application of artificially inoculated barley seedlings is that by applying ethylene, we not only have a reduction from the beginning in the disease symptoms itself, but we get a similar picture with regards to the fungal growth within barley. And this is quite interesting because it means that ethylene not only reduces the symptoms, but it also inhibits the growth of Rammelaria. And therefore, this is the reason why I want to investigate how different barley varieties respond to ethylene. And therefore, I've identified 2 marker genes I want to test soon, and hopefully in the next meeting I can show you those interesting results, because I'm quite excited to do the next experiment. But with this, finally, I want to acknowledge all my collaborators at James Hutton, KWS, SHRUC, the Scotch Whisky Research Institute, who's also providing me with funding, obviously the International Barley Hub, RISAS, but also the BSPB, who kindly provided me with 3 extra studentship months at the end of my PhD because of the corona pandemic. Without them, I wouldn't be possible to do the exciting ethylene responsiveness work. And with this, I want to thank you for your attention. And if you have any questions, even later in the day, just email us, contact me on Twitter or LinkedIn. And thank you so much.
Thanks very much, Laura. That was brilliant. 3 very complementary presentations there. So giving comprehensive look across barley, and in particular, and some detailed aspects of Rammelaria and its control and mechanisms of disease. So shall we go to the questions then? Robbie, would you like to ask your question to Francois?
I thought you were going to read it out, Adrian.
You can express it so much better yourself.
Yeah, Francois, I was just wondering whether or not the pattern of rubellin pathway gene expression that you showed was consistent with the development of disease symptoms. And then secondly to that, is there any evidence at all about a causal role for rubellins in Rammelaria disease progression? And the third part of the question is, if there is, what could the on-off switch be? Because there must be an on-off switch, I presume.
Yes, I can sort of answer the third question to start with. So those genes are, there's a lot of regulators going on in secondary metabolism in general. And you've got 2 layers, if you want, of regulation. There's one layer generally that occurs directly at the cluster level. And that's the transcription factor I mentioned. There's one of them. There's potentially 2 of them involved in the regulation of this cluster. But these tend to be under the regulation of master regulators of secondary metabolism in general, and these are under they're basically responsive to pH, for instance. So I've investigated a few of these master regulators, and the PAC-C, which is the regulator linked with pH, seems to be following the same trend in terms of expression. There's also a nitrogen supply as well, can be perceived, and that also acts as a switch on-off, but at the literally not just the cluster level, but completely changes the second metabolism. And light as well is known to affect that. So there's what's called a velvet complex, which is a complex of 3 different genes that act together and that can switch on and off those transcription factors at individual cluster level. So in terms of the links between rubling production and symptom development, it seems that the toxin is, or at least those genes are switched on really before symptoms appear. And as soon as they start appearing, then they get switched off. The difference is that we don't really know, and something that we need to do is to quantify in the leaf when we see this toxin being released, because there's a difference between the production and the release. We know that the fungus is able to store some secondary metabolites in vesicles. They've been seen actually in the fungal hyphae as red vesicles. So there is a red compound, potentially could be rubulin. It could be some intermediate. It could also be something else. But that's something would be interesting to see, is when exactly is it released in the leaf. And that would help us understand more the link, if there is, between rubulin and symptom development. Having said that, I've done some infiltration on barley, and particularly on barley that is on different types of barley, so resistant and more susceptible varieties, and also land races as well that we knew were from field data were more or less resistant. And it doesn't seem that there doesn't seem to be a correlation between resistance to Remularia and sensitivity to the toxin. So it's not as clear-cut as if a variety is sensitive to the toxin, then it's susceptible to Remularia. So that link doesn't exist really. So again, raising the question of what really that toxin does. It could be involved in basically fighting off other pathogens as well, protecting its ecological niche. That's a very strong possibility. But because it acts as well, it induces cell death, it must have, or it's likely to have, some form of impact on disease development. But whether it's a pathogenicity factor that's required for disease or a virulence factor that just increases the severity, that's something we don't know at the moment.
Thank you.
I'll ask Robbie's next question for him, because he brings up MLO, which I thought would come up when it comes to anything to do with Gramularia. Is it within the confidence interval of the 4H peak that you found on your GWAS? And is it associated with susceptibility or resistance? I know there's been quite a lot of controversy over this over the years. So, Laura, got any thoughts on that?
You're muted, Laura.
Muted, Laura.
Sorry, Adrian. Have you just read out Robbie's?
Yes.
Yeah.
Okay.
Yes.
Sorry. I read Joanne's message in the chat, and yeah, no worries.
We can move on to that straight afterwards.
So the second part of the question, so yes, we know from several different studies that MLO is associated to increased susceptibility of Remularia, although there's a mixed picture. However, I've done quickly with Mark Loosely, who worked for the James Hutton at the beginning of my PhD, if MLO targets the end of the chromosome 4H, and we did find that is like it targets the confidence interval of my QTL as well. But to be quite frank, I've not really looked into this after this because my bioinformatics skills are beyond this, and I struggle in using all these data platforms available for Barley. And yeah, but we know there are associations between LMO and Remularia, and it's definitely one thing I have to look into before I finish. but it's difficult, I guess, as a PhD student, like asking for help all the time, which I'm trying to overcome day by day.
Laura,
But
as a PhD student, you're responsible. You have to ask for help every day.
exactly, yeah.
Yeah.
Would you like to comment on Joanne's point as well?
Yeah, I hope we can come up with a new project to tackle Ramilaria, but it's probably, yeah, a question of how and what to do. I guess it depends on who's involved and who wants to be involved, and yeah.
Okay.
I think there's
Another-
definitely potential there to take the project further.
And another question to you from Matthew Ruscu. Laura, assuming leaf RNA-seq is available, it should be very straightforward to determine the status of MLO. And they have data on Aramir.
Oh, okay.
So
That's
more of a
good to know.
comment than a question there. Yep.
Thank you, Matt.
Yep. Actually, could I ask a question? It could be any three of you on Ramilaria. We're focusing very much on symptoms, and they're a bit notoriously difficult to both assess and reproduce and things like this. How well correlated with this focus on symptoms, how well correlated is that in terms of the actual damage it does and the actual damage it does to yield at the end of the day? Are we focusing it in the right way here for control of Ramilaria? Any of you for that one?
I think the paper, the first qPCR paper that we produced, demonstrated a clear link between pathogen DNA and the symptoms. But I think Gareth Hughes did some work on a big data set looking at Ramilaria symptom development and yield loss, and we did see that there is a significant impact. So the more symptoms you get, there's a significant impact on yield. Now, Ian Bingham had a PhD student, so we were looking to see if there was any effect of pre-symptomatic infection of Ramilaria on barley yield, and she couldn't find anything clearly. So I talked about, barley being source limited. Didn't seem that early infection had an effect. The yield losses that she did associate with Ramilaria were primarily down to loss of photosynthetic area, loss of GLA. So that's why we focus on that. That seems to be the important aspect.
Okay, thanks. Henrik, you have a hand up. You'd like to ask your question?
Yeah, related to that, so photosynthetic laws now, this is a photoactivated toxin now. I think, as I recall, that there are reactive oxygen species involved, and if so, whether they target the photosystems.
So the Rublings, for sure, they've shown, well, all the work that was done on the mode of action before was done in vitro, and they showed indeed ROS production and associated that with membrane damage rather than any impact on the photosystem. They've not really looked at that, to my knowledge anyway. And yeah, again, it was only done in vitro as well. So they were just basically literally putting in vitro some components of membrane and with ROS. And yes, obviously that was going to create some damage on the membrane. That wasn't too surprising in itself, I would say. But it's a good point, yeah. It'd be interesting to know if there is an impact on that.
Thanks.
Maybe I can have a question. Thank you. For Laura, for the ethylene study now, I think you mentioned senescence. The question is whether ethylene promoted senescence. You nicely showed that ethylene suppresses disease, but wouldn't it promote senescence? And the other action that ethylene has is also promotes defense. So it's probably difficult to separate the two unless you look at possibly target genes, etc. But even in that case, it might be difficult to untangle the two. Yeah.
Yeah, thank you for your question. So yeah, so the reason why I used ethylene in the first place was because I wanted to enhance senescence. But looking at my spot data, it didn't really do the job it was meant to do. But because we got so interesting results in terms of symptom expression and inhibiting ramillaria growth itself, I went on with the concentration. And yes, and ethylene is a defense response, or is involved in the ethylene in the immune response of plants in general. So this is kind of my explanation why it could help barley to reduce ramillaria growth in the plant itself. And this is a reason why I want to have a look into my different into my different spring barley varieties, if there's a difference in the ethylene response that can be then related to my symptom expression in general. So if there's a differential response of resistant and susceptible varieties, would this explain the mode of action of ethylene, why it reduces the ramillaria growth in my experiment? But it's like very early on data, and yeah, it's a very interesting project, but it will be very difficult to combine this with ramillaria itself. So this experiment I'm trying to do on the ethylene responsiveness will exclude ramillaria, because it's a pathogen which then also induce ethylene in the plant. So it's then very difficult to untangle those two. So I will first try to see how my barley varieties respond to ethylene in those ethylene marker genes, to see if there's a differential expression in those marker genes. I hope this answered your question.
Oh, thank you.
Thank you. Can we just return to this question that Robbie asked earlier on, actually, which is from, first of all, says an excellent summary to Neil of all the barley diseases. From all these biotic challenges, which one would you prioritize for finding a genetic solution? And do you know what type of resistance or tolerance is available in the gene pool? I was actually going to ask also, what's the next big disease that you think we should be focusing on, which is related in a way? You've obviously highlighted things like rust in terms of climate change likely to increase, but maybe there's other things as well. So what sort of target should we have for genetic control in future?
Okay. Thanks, Adrian. You're not going to be surprised that the answer to part A of that question is going to be ramillaria, obviously, because because we're talking about the pathogen, as Laura highlighted in her talk, where the big issues about chemical control, which we've relied on far too much. And if you look at the breeding effort, which has gone into control, the other pathogens I talked about, there's been a lot more effort than there has been in terms of ramillaria. Funnily enough, that they talk about MLO, there is a theory that the introduction of MLO has exacerbated the symptoms we see from some of the necrotrophs, so the Rincusporium, net blotch, and ramillaria. So whether perhaps we need to think about giving up this complete mildew control and looking for a more robust package is a good question. I think Laura's answered how much genetic control is there in the elite lines that we used in the Coracle project. So there is some variation, but not a huge amount. We may have to go back to some older germplasm for that. In terms of what's coming next, well, nobody saw ramillaria coming, did we? In terms of change to the climate, I think you've got to be looking at pathogens which our predictions are for, at the moment, wetter winters, wetter autumns, and then possibly warmer summers. So that would favor things like the brown rust, although we haven't really seen a major brown rust epidemic since 2014. So I think the rusts would be something to look at as well. The effects of changing practices and shortening rotations is going to have implications on a lot of these stem-based diseases as well, and things like fusarium, I think. But some of those minor ones, I mean, ramillaria was only first UK report was 1998. And here we are 24 years later, and it's a major pathogen. So it may be something that we may have observed and went, oh, there are many other pathogens I didn't talk about, really, well, small ones, halo spot and things like that. They may become more of an issue going forward.
And do you think changes in farming practices or trends in farming practices going to favour any particular diseases? Because quite a lot changing in the way we're doing our farming at the moment.
Well, the way we're going, there'll be no more nitrogen going on crops because nobody will be able to afford it. So that could have a big influence. Yeah, you're right. Changes in crop architecture and crop canopy, that will affect microclimate. So things like some of the biotrophs might not like that so much. We'll be looking at the splash-borne pathogens and the cool wet ones, I think, will be the ones to keep our eye on the next few years.
Okay, thank you. If I've missed anyone's question,
I think I
shout
saw
out.
a hand up from Ignacio in Argentina. I know he's up early this morning. No,
Right.
I thought he had his hand up.
Didn't see that, but ask away if you've got something.
No.
Go for it.
I didn't.
Oh, you didn't? Oh, I thought I saw your hand up. That's okay. Just checking.
Okay. Well, in which case, if there's no more questions, it just remains for me to say thank you for 3 excellent talks, even if not quite in the order we planned in the end.
No.
That didn't really detract at all because it covered a wide range of things and some quite detail in what maybe Neil regards as the most important disease at the moment, but certainly a disease we need to constantly keep an eye on and help to combat. So thanks ever so much to all 3 of you for your contributions. And in fact, we've just now got the slide up for my next point. So I don't even have to look up the details because they're there on your screen, which is the next speaker in about 2 weeks' time, I think it is, Laura Rossini on the genetic control of plant architecture in barley. So hopefully you've already registered for that, but if not, please do. And we'll look forward to seeing you at that seminar. So thanks very much.
Okay.
Yep.
Thanks, Adrian.
Thank you.
Thanks for
Thank
hosting.
you.
Bye, Robbie.
Thank you. Bye, everybody.
Bye.
Bye.
Thank you.
Thank you. Bye.